Hello, and thanks for joining us for the Spatial Biology Revolution 2023. My name is George Hammond, and I'm here to moderate today's event alongside my colleague Princess Senate. Before we start the talks today, I just have some general information and a few tips for getting the most out of the event. We would like to ask if you could please refrain from video, audio, or photo recording of all talks. Many of our speakers have kindly agreed to share their slides with you, and these are available within the literature section on the platform. Please remember that we encourage debate and discussion and welcome all of you to use our networking services to introduce yourselves to other attendees. But we do not permit the overt solicitation of products or services and reserve the right to block any user who infringes on this rule. It's also incredibly important to say that this event was only made possible thanks to the generosity of our sponsors and partners, particularly our Technology Spotlight sponsors, 10x Genomics, Canopy Bioscience, Leica Microsystems, and Miltenyi Biotech. For more detailed instructions on how to make the most of the features available to you at this event, please navigate to the event home and watch the overview video at the top of the page, or visit the FAQ section in the left-hand menu. And with that out of the way, I will now move on to introduce our keynote speaker, Dr. Jasmine Plummer, presenting Spatial Biology, Is It Really Worth the Hype? Dr. Jasmine Plummer is the founding director of the Center for Spatial Omics and member of the Comprehensive Cancer Center at St. Jude's Children's Research Hospital. She received her PhD in molecular and medical genetics at the University of Toronto, Mount Sinai Hospital. Dr. Plummer's lab uses a multi-omic approach for single-cell and spatial genomics to better understand the pathogenesis of neurodevelopmental disorders and cancer. A very warm welcome to you, Dr. Plummer. Following the presentation, we will have a Q&A session, and we welcome any questions you may have. Please ask questions using the Q&A system to the right-hand side of the video player. And now, without further ado, I will hand over to Dr. Plummer to begin her presentation.
Thank you, George, and thank you to Technology Networks for this wonderful invite to kind of give you a really high-level kind of overarching approach. I hope we'll give you a tour de force today, walk you through very specific examples of how we think spatial biology is coming out and emerging as a field and is really worth the hype and why we're all on this call today. So one, thank you for attending, and hopefully we'll achieve our goal of being able to see whether or not, why even use spatial? Is it just fancy marketing, or are we in a new age of discovery? So my goal by the end of this is to hopefully achieve that you will think by the latter part of this that we will be in the spatial realm. And so I really do love this slide where we talk about the idea of using things like bulk transcriptomics, right? Where it's a smoothie analogy, where we took everything in the cup and we mushed it up, and we got some really interesting information. The TCGA, the Cancer Genome Atlas, gave us lots of interesting information to understand how cancer was progressing, more outcomes. And then the last explosion in the field was single cells. So we've been seeing a lot of single cell genomics occurring lately. And that really is to say we can now identify the raspberries, the mangoes, the pineapples, the kiwis, and the blueberries in the smoothie. But what I hope you can believe is that really where we are right now is the right-hand side, is that spatial and really the condition of actually most of our biological tissues are not in a red versus blue versus green, but really the intermixing of how these fruit are coming together, or really in the case of this. So I love this slide because this is the truth of what we're talking about. When we do bulk tissue of anything, whether that be DNA or RNA sequencing, we really have mushed everything up, pelleted it down, and put it on a sequencer. And what those single cell maps actually show you versus the red versus the blue and the green, it really is still that we mushed up the entire tissue and put it in a tube. But now instead of a big pellet at the bottom, you can actually see it in a single cell tube. But here is really where I think spatial is. So on the right-hand side, we can see for anybody who is not a biologist, the top is cancer and the bottom is normal adjacent, meaning this is breast cancer in a woman that for 40 years we've been able to diagnose by just putting a $2 stain on a slide. And for the regular eye, you can see these are two very distinct things. I don't imagine that if you were to put a single-celled tube of a tissue from a tumor versus normal, whether or not you'd be able to distinguish it. And we can tell you that this is real live data in the sense that we have single cell breast tumors here that we've disassociated. We can see all these nice populations, but I really want to show you is the spatial RNA-seq. So this is using the Visium assay. And what you can see is that regular slide, right? Remembering that we are staining this with H&E. And on the left-hand side, we can see all different parts that a pathologist can see, but really at a sequencing level, take a deep dive into these individual cell types that we would have seen in the single cell, maybe projected on a map, but now we can actually see into the tissue itself. And what does that mean? So this is a real life example of those maps, right? So you have maybe a map here of 63,000 cells. We find maybe interesting cell populations. Maybe this is a cool cell population and this is another cell population. But I really want to pay attention to is these 44 cells. So in the single cell data, we would never have seen these cells. And we know this because we run many single cell experiments. And we can tell that we've learned a lot from it. But those 44 cells in a breast cancer tumor actually are in this lobule. So they really denote a particular part and feature within the breast cancer itself that we're not seeing in the single cell data. So we know from this, hopefully, you can see that spatial biology adds another dimension to these small populations of cells that we're not able to see in the single cell data. And actually, it's how these cells are organized, right? Why is this red here versus the green? It's really how these cells interact with each other that actually is the reason for most disease. And why you're probably on the call is this slide here. And so we know we're taking a tissue section, we're spatially resolving it, and the hope is to be able to look at genome, epigenome, transcriptome, proteome, and look at it the same way that we have done in both bulk and single cell data. And on the right-hand side is just this beautiful review from Chris Mason's lab that actually talks about the explosion in this field. So it doesn't even demark 10 years here, but what you can see is like there's been this many. Most of these have actually lived. So the blue have lived mainly primarily in research labs. What you can see now is that there's this huge adoption now in the field that really has really augmented in the last few years with the commercialization of the equipment. And what we'll really focus on today is a lot of the RNA and protein, but I really want you to see in panel B here, where most of it is really developed in RNA. Some is kind of slowly growing in protein, but very little has been done on the DNA aspect. So I think that is one place by which we'll see a large explosion for the next half. So let's walk through the actual case study. So our first one's going to be kind of a multi-omic approach that we've taken in neurodevelopmental disorders. And if you can see kind of in this Lego diagram of our smoothie analogy, I would hope that everyone would understand that in the brain, it really matters where the green cells are versus the blue cells, because we have a higher order of magnitude of understanding and making those cortical connections. And so one of the things that we work on in our lab is really studying kind of the neuropathogenesis of some mental disorders. And so why do we care? About 25% of the world actually exhibits some version of a mental health disorder. And kind of the genetics of this have been very poor. We've understood it for a long time, but we don't have genes the same way that we have in kind of cancer. But what we do know, if you look at this map, that yellow is unaffected and the orange is affected, is that there is some genetics to it, that you do find it through generations in a family, but it doesn't usually show up in the same disorder. So you may have schizophrenia as one family member, but autism in another. So we're really trying to find how are these connected. So if you look in bulk data, if you were to take an autism person and a typically developing person, what you would do is we take kind of the places in their brain that we know these disorders to affect. And what I want you to see in the bulk data is in typically developing places, we have genes that define kind of the frontal cortex and the temporal cortex. But in autism, those genes are all array. So we really don't have that nice patterning of transcriptomics or gene expression that you would expect to say, this is the part of the brain that is defined by these genes. And it gets further complicated by the connectivity. So when you look at ASD or schizophrenia, you can see in these kind of diagrams that the way that these kind of neurons are actually connected are very, very different. So obviously spatial is an approach by which we can take this map of this single cell data, remembering, right, that these are all cells that we've mushed up from the brain. We can find all these very cool cell populations, neurons that we know about, glutamatergic neurons, radioglial neurons, and then we take the approach where we actually take the brain and we can map it. So we've taken pieces of the brain in places that we know are important for neurodevelopmental disorders. And what I really want to show you very quickly is that from this single cell data, when you're able to pair this kind of multi-omic approach, while we don't have single cell in this is Visium assay, we can actually get closer to single cell data by kind of fusing the two and merging them. And what does that look like? So when you look at those single cell populations, remembering like this was a cell population that was orange in a UMAP. But what you can see is it actually starts to define subcortical layers that we weren't able to see in the single cell data. So what you can see here is like glutaminergic N6 down in this panel is actually highlighted through the whole subventricular zone. But if you look at glutaminergic N3, it's actually once a neuron becomes kind of a neuron and hits the outer cortical layer, then it turns on these kind of markers. So as you can tell, even if you're not into brain development, you can see that these maps, right, that I'm guilty of generating a long time ago, is that all those cells now have a context to who they are and where they are. And I'm going to walk you through a very specific syndrome of why this becomes important. So TCF4 is a rare disorder, and so it's Pitt-Hopkins, but it's also a gene that's characterized by intellectual disability, developmental delays. It has very stereotypical phenotypes, but it's also being found as a gene in general GWAS for schizophrenia, and is being linked as an autism gene. And I love the idea of thinking of multimodal. So spatial isn't the only way, but we can kind of use all our old data and merge it into kind of the spatial data as well. So this is data from the Allen Brain Atlas. And what I want you to see is that there's a very typical expression pattern of TCF4. Everybody knows this gene because it's a master transcription factor. It gets turned on in the brain at high levels and very key places. But what you'll see here is we found this reciprocal transcription factor that we wouldn't have seen actually unless we looked in the bulk data. And you can see it beautifully mimics the opposite of TCF4 expression. When we look in our single cell data, we actually see TCF4s everywhere. It doesn't define one particular cell type because we know that it's a master transcription factor. But when we looked at our SOX9 data, we actually didn't see anything super interesting in the single cell data until we looked at the bulk. So now we can start to see there's these kind of two cell types that we see, but most pronounced in the spatial data is where we find the difference. TCF4 is everywhere, except it doesn't get turned on except in a very high level once it becomes a neuron and only in certain neuronal populations, but it's an exclusion rule. Wherever TCF4 is, and we've done the statistics to prove this, SOX9 is not. And wherever SOX9 is, and so now we're starting to get players to kind of these higher patterns of what these master transcription factors are doing. And we would not have seen any of that had we not looked into the spatial. So I think that I've shown with this particular example that context really matters. So we kind of walked you through, right? I love this. This is the human cell landscape. It's every single cell in the body. But the reality is like those cells actually have to give definition to the body as the organs are developing. And I think cells also, what we've learned in biology is they also feed off the environment they're in. So, you know, an epithelial cell in a heart looks very different than an epithelial cell in your skin. And one of our other applications through collaboration with Dr. Harry Fu in Ohio State was to apply it to Alzheimer's disease. So not only have we applied it to normal human brain development, but we've also applied it to the disease state. So what you can see here, kind of using the same approach with Alzheimer's disease, is we took both single cell biology, paired it with Visium spatial biology. And what you can see is kind of, I depict this control versus Alzheimer's. What we can see is that we actually found this astrocyte-specific gene signature that we weren't able to fully detect in the single cell alone, that now we can start to detect in the Alzheimer's. So you can see all these now, these white puncta that you weren't able to see in control brain, but now you see in Alzheimer's. And here's the statistics to kind of prove that we are seeing a difference. We are kind of taking this and venturing it into other biologies. And so I really want to talk about a little bit of the spatial that is not just Visium and single cell, but we are using things like genomics by nanostring, where you can pair kind of proteins against the whole transcriptome. So we don't get single cell data, just like Visium, where we weren't getting single cell data. We can pick, and I love this picture because it's 192 patients. You can see we stained for green and blue. What are the green and blue? Well, the green and blue are stroma versus epithelial. And so I can imagine that when you mush this up, right? If we were to do this in the past and we mush it up, we wouldn't have this beautiful array to show us that every tumor looks different, even by these two markers. So imagine when we pick green, so we pick stroma versus epithelial cells, we can actually see the tumor microenvironment and cell type specific changes even at this level, and now we've paired it to bulk RNA-seq. And this is through a collaboration with Dr. Sophia George and her pathologist at Sylvester in Miami. So kind of now I think I've given you proof of principle that single cell bulk and all our bulk technologies can really be fused into spatial, so that spatial doesn't have to be a standalone, but it's really giving us information that we were not seeing in the past. And walking you through not just normal development, but disease development, and taking you all the way through not just Alzheimer's, but into cancer. So now I want to walk through another case study. Now that we've been through four, this will be our fourth one, was really to think about applying spatial omics in a way I think most of us have not been able to make a full penetration because of the issue of health disparities. Because in health disparities, there's so many things going on that just isn't case versus control. And so how have we been doing this? So this was really spurred, and this kind of is something that keeps me awake at night. So I like to give this, and I hope everybody on this call will go and kind of take a look at this graph, because we're all on this call for this reason. There has been an explosion of genomic studies, and this shows that as of 2021, so if you look at this, it's plotted from 2006, and we've had this explosion of genomic technologies and genomic studies. But we can see that the vast majority, 86% of these genomic studies, when we look at the global population here, color-coded. If you take the red population, that's of European ancestry, and you can see the biggest demarcation of all those studies, which 86% of them, have actually been in European ancestry. Despite 81% of this explosion in genomics, we're really only covering a very small percentage of the global populations. And so there have been very few studies that have been conducted in underrepresented populations. And so we've really tried to expand this in collaboration with the AC3 and Dr. Sophia George's group in Miami to really look at, and we'll walk you through our case study of breast cancer, but really trying to look at like why these health disparities are occurring. So we know that normal tissues in Black individuals are very understudied, but they have a disproportionately more aggressive disease. They have higher incidence of mortality, and their disease is actually refractory to treatment. And we don't know if that's the biology or really that the treatments haven't really been made for this group. And what I want you to see on the left-hand side is we're really interested in looking at African ancestry. So when you plot African ancestry, you can see that they're in the green here. So they have actually a lower incidence than America. So they kind of have a low incidence, but a very high mortality. So that was often thought because it was due to access of care and that, you know, maybe they had a late stage disease. But what was really interesting, so the blue here is we know we have better, probably likely better treatments in the US, so we have a high incident, but we don't like die as often in this population. But it's really interesting when you add this admixed population of the pinks. The pink are African Caribbean. And so when you add that population, you can see that the African ancestry really in terms of mortality and incidence are actually all over the map. And when we look at that population, they also have access to care issues. So there is something going on here that is very unique to the Africans, the US Black women versus the African Black versus the Caribbean Black. And so we really took a pronged approach to really invest not just the scientists, the pathologists, and we worked with Akoya. So a big thank you to Nadia and Dr. Ben Sheikh there who did the analysis, where we paired with pathologists at Sylvester and broad across the globe to really develop a panel that we knew was going to be clinically diagnostic. So you'll see diagnostic markers on this. Then we also asked them, well, one, from a pathology level, what would be the things that you would really want to see? And then we also mined our single cell data to really think about the cell populations that we need to find or would like to find in these cancer types. And so what I want to show you is that this is the difference between the single cell approaches and the approaches that we're using now spatially. So if we were to talk about just a cell population level, it's very hard to find things like CAFs, because if you look at all these cells here, so I can tell you there's about 700,000 cells, I think, on this one slide of tissue. And we can see if you just look at the yellow, I can tell you that yellows are stained for CAFs, so cancer-associated fibroblasts. We know that these are often associated with more aggressive disease. And so we actually don't detect this in our single cell data, because if you were to take the proportion, when we look at single cells, we're taking 10,000 cells, let's say, out of 3 million cells of a tissue. And so the largest proportion that we're going to profile are going to be these kind of cyan cells. And maybe in this tissue, because it's a very aggressive tumor, we're going to get all these kind of well, actually not aggressive, we get CD8 cells. So we have like this immune infiltration. But I want you to see most beautifully in these tumors now that we can see is we can see these calves. We see those calves are highly infiltrated by CD8 cells, but there's also calves over here that are not. And we know that there's probably a biological reason for that, but we were not able to detect it in our single cell data. So how do we actually look at this? I will say that it is, for all that everybody thinking about this field, I love to start with this slide, is that there are going to be computational challenges with the idea of this big data. So again, back to this slide, when I said we couldn't detect these because maybe they were 0.1, they're 1% of this entire slide, if that. We are probably processing 1,000 to 10,000 cells for every sample. Now you can imagine we can process up to 400, 500,000 cells per sample. Not only do we have to process these numbers of cells, which are giving us more biology and more insights into the data, it's also a huge bottleneck to how you interpret the data, because we also have to process these 500,000 cells and give them a spatial coordinate. But how do we do this? Well, I can tell you we do it quite well. You can see here that we have a tumor versus normal. I think this is a beautiful example without any of the markers here to say we can take these tissues and really truly look at the definitions of what a normal adjacent part of the tumor and the tissue itself. And what we do is all the single cell approaches. You essentially define the cells, which you can do quite well, put those dots inside the cells. So these are dots. These are probe sets. So I'm actually showing you a breast tumor from a Bohemian woman that was actually run in another type of technology, which was VisGen technology with RNA-ish probes. You can see that we can actually map them into dots. These aren't actually the same sections, but I wanted to show you the idea of how you take those probes and you put them into dots. So this is kind of where we're at. I will tell you that we're going to be up to 8 million cells soon. And so in last year, a Goliath effort, which was the Human Cell Atlas, Tabula Sapiens, was a multi-organ across all single cells with 1 million cells across 24 organs. A beautiful lung atlas was published just this past June with 2.4 million cells. And now you can see my computational problem. I have 4 million cells and I'm not even done this the study yet. But you can see we can see the same kinds of things that we see in the single cell data, even with just 66 plex, because those 66 plex were chosen from single cell data and from kind of previous experiments where we knew what cell types we wanted to look for. And we can tell you that we really can see ancestry differences that we were not seeing in our single cell data. So we can, the right side shows a heat map. We can find all the kind of cell markers that I denoted from that heat map. What we can also see is in that, and even in the biopsies, we can see the cancer epithelial cells versus the normal epithelial cells. So we can actually distinguish cell states there. And what you can see here is we're staining for very principal markers, E-cadherin, and immune cells. And what you can see here is that you see kind of an immune environment difference between the US Black and the Caribbean Black. So remember that, right? We're staining for the same markers. These are biopsies. I'm showing them for a reason too, because we can see biopsies as well as tissue, which becomes very important if you want to apply these to real life clinical trial samples. It was important for us to establish this metric. But even in the biopsies, you can see that there's a whole different component of CD45 in the Caribbean Black women versus the US Black women. And we can see this really when we take deep dives. We can see immune cell neighborhoods are altered based on their ancestry. So you can see here we're staining for E-cadherin, which are the epithelial cells. And you can see that, that we have very much these immune infiltrations in different environments based on the ancestry. These are very hot tumors, but even in the hot tumors, we see different vascularizations and different phenotypes for cell proliferation. And how do we do that? So we've been taking a machine learning approach to this. And so what I can tell you is there's lots of different ways to look at neighborhoods. So you can do calculations of like Does an epithelial cell tend to live around vasculature? Does an epithelial cell tend to live around immune cells? And so we've identified about 12 neighborhoods. So these neighborhoods of what these niches look like. And you can tell that these neighborhoods, they're shown here just by different colors. They're very different proportionally. And we actually get some neighborhoods that do not exist in the US Black that exist in the Caribbean Black. And you can see that we've done a lighting clustering here just to show you that in those biopsies themselves, you can see that those neighborhoods look very different. So these aren't just the cell types. These are the cell types of who they associate with. Is an immune cell mainly around an epithelial cell, or M2 macrophages mainly around epithelial cells, and we're seeing those differences amongst our ancestry. So can we do better? I say this because this is all learned, right? So we have to go in here and tell you exactly what cell types we want to see. So it goes all the way back to here, where we've assigned cell types based on the idea that you have an EK adherent stain, so like your epithelial, you have a collagen 4 stain, so we know you have some level of a fibroblast component. What kind of fibroblast are we going to name you? These are all, in my mind, still quite arbitrary in terms of what we're labeling them. So we've been trying to really think about, what are these tumors telling us? What is the underlying idea of why the US Black tumors are very different from the Caribbean Black tumors? Because we can see different neighborhoods, but agnostically to what the cell type is, why are these neighborhoods different? So we've been taking some machine learning approaches to just try and learn, are you more oncogenic? Do you have more cancer aggressive features? And so we've paired with Dr. Malta in Brazil, who has taken the idea, who's taken a machine learning approach to the idea in the field that stemness, this cancer stemness, right? So you have these stem-like cells, they become very resistant to therapy. And those are the ones that are known to kind of see recurrence. And maybe that's why using the idea that cancer stemness is really the aggressive parts of the tumor, are those cell annotations really being dictated by the stemness? And so we took her kind of bulk, this was made in bulk RNA-seq data, methylation data, the cancer genome atlas data. So what you can see is she, even in breast cancer, using her algorithms can really define normal versus really aggressive tumors in the TCGA for breast cancer using this algorithm. And so we can really distinguish kind of cancer stem cells, even using staining. So you can see the blues versus the reds. But we tried really the first approach was just to say, can we do this at a single cell data level? So for any kind of machine learning or AI tool, you need a lot of input into it. So in this, we actually used the single, instead of the cancer genome atlas had a lot of patients in there, and they used that to learn the tool. So what we did was kind of learn the tool by all the cells in our data set. So we took every publicly available data set for breast cancer, and we included it across all cell types. And so what you can see in this UMAP, we're able to find kind of all single cell data that you expect. What I wanted you to see is our tool. So what our tool actually does is stemness is red, high stem is red, and low stem is blue. And what you can see is in these, they pair very well with the more aggressive cell types. And more importantly, they actually pair quite well with the clinical subtypes. So in this case, we know that triple negative breast cancer, so the green is the most aggressive, so most stemness-like subtype. And if you see here, the green's kind of here in the clinical subtype, and what you can see in the stemness index, those are the cells within this data set that pair very well to the most aggressive form of the disease. And we would like to say that this approach is agnostic to whatever spatial data set you can throw at it. So we paired it, we didn't show it to the Visium, so it works in Visium because that's a very easy pairing and from single cell into Visium. But you can see we've paired it into both all three RNA-based approaches and protein approaches in PhenoCycler, CosMx, Xenium, and Merscope. So in this sense, you can annotate the cell types the way you want. So we've taken the annotations across all the data sets, and you can see that the stemness tool, both in Xenium, you can see within that breast cancer, we can see the most aggressive cells within that. We can see that in this VisGen data set, this is what really beautifully to see, because remembering that we can see the normal in this data set versus the tumor. And you can see even with normal, we don't get any cancer stemness, but only in the tumor. And in some populations do we see it. And the same for the COSMICs data. So in the COSMICs data, where you see the most epithelial tumor annotated, you have the most stemness cells. And what does that mean in our actual data set? Here's a beautiful representation of that. So here we have kind of these high stem cells predictions. So this is where we were before. You remember I said, I really want to get away from the idea of just knowing the cell annotation and me labeling my epithelial cells versus other cell types. What you can see here is that our data pairs very well to the idea that this biopsy, ignore what the cell annotation is. This biopsy has stemness throughout it, so it has cancer stemness. What you can see in the US Black versus the Caribbean Black women is that in this case, there is stemness that is very, 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 very high in certain regions of the Caribbean Black that we do not see in the US Black women. So what I want to end in the last kind of 5 minutes is to really think about kind of, we talked about, is it worth the hype, and kind of where we're at. We're kind of at the beginnings with these spatial transcriptomic approaches, kind of venturing now. I hope I showed you some beautiful data into some protein approaches. What's that runway really look for us? If we're all invested in the idea of spatial, kind of how are we thinking about this? So from a St. Jude perspective, we've started part of our strategic plan, which was $2.9 billion over the next 5 years, was to not just do this in a adjacent way to single cell data. We really wanted to take an approach by which we had our whole hat into the idea of what spatial was going to be. So we've started a center for spatial omics. And this center's investment is across all spatial technologies, albeit whether that's multi-omic, whether that's RNA-based, whether that's sequencing-based, and whether that's protein-based. And so we've invested in them all. I've given you examples of some of the things that we've looked at. But most importantly is that there's infrastructure to support those growing needs. So this is the team so far, but really, in my opinion, most of the team really is going to be, maybe when I first projected, a 1-to-1 ratio of like laboratory to computation. But really we can see that, we live on a fiber optic network floor. Most of our investment, the first piece of equipment I actually did buy was my own GPU and CPU, despite the ginormous resource that we have here at St. Jude. And so we really are applying these basic research models, both at a very basic research level. So there are Drosophila people here at St. Jude, all the way to kind of why we are showing you those biopsies. Of course, we can do these things in FFP and sections, but can we do them as a patient is going through clinical trials? And I think I've shown you data to say the answer could be yes. So the future for single cell and spatial applications, given I've given a lot of talk about health disparity, I wanted to give a very quick plug for us trying to make this accessible to everybody. So Dr. Martoletto has been working with myself and a larger group on kind of taking these blocks, so taking your FFP block, nuclei extracting it, putting it onto single cell tools, and really being able to take blocks of single cell data into tissue. And so we did this across it's out for review now. We did it across many different normal and tumor tissue types, both in single cell and Visium. And I just want to show you the data is very good. So we can take these tissues, we can make it into all those cell types that I showed you in earlier slides. So not only are we invested in the idea of something spatial, but if you are invested in single cell technologies and you want to open your blocks, please, there's a bioarchive, please look for that. We do think that there are ways by which we can move those needles forward. So I think I accomplished my goal today, which was, do we think these applications can apply across technologies and disease? I would say yes. Not only, you probably thought it was a spatial top, but really even saying that we're still invested in single cell. Can we pair the single cell into spatial, even despite using blocks that are nuclei based? I would say, is it just fancy marketing, or are we in a new age of discovery? I think the verdict's still out. I think we need more data, more QC, and better computational pipelines to do that. And so this is another plug for the idea of kind of data standards. And there's been a call we've started a group called the Global Alliance for Spatial Technologies. You can reach out to Dr. Marletto or Dr. Vlachos or myself to kind of be included. But the idea is to really have more vetted QC, both in laboratory and computational. What is a cell? My group's really struggling with the idea of proper cell segmentation, how to assign transcripts or protein into a cell. Who's going to establish these? Hopefully it won't just be us, but we welcome everybody's insight. And I think it's a very important idea is that where are we headed? So when I was talking about spatial, I think I've shown that we can identify new cell types linked to the pathogenesis of the disease, but multi-omic approaches are required to better understand them. So we can't just throw our hat and ignore our old data. That it's really important for researchers to bridge the gap between clinicians and pathologists to better interpret, because it's for the first time you really have to sit there and look at the tissue instead of mushing it up. And that computation is the way, hence this. Is data science just a buzzword? These large data sets require better processing, computation, and analytical infrastructure. But I think, and I'm really optimistic, that machine learning and AI will help with that computational burden. But again, back to my plug of more standards and more QC and more data, the more data sets we have to learn and test and validate these models, will be in a much better place when I have to give this presentation again. Hopefully I'll be invited back. So this is an acknowledgement slide to all my funders. Some of this work was done in my previous institution at Cedars. A big shout out to the AC3, which is a global consortium, as well as the Transatlantic Gynecologic Cancer Foundation for some of the access to the tissues, and all the neurodevelopmental work with my colleagues down there and our funding sources. So thank you again for the opportunity. And I think it's time for me to welcome some questions.
Thank you so much, Dr. Plummer, for that fantastic presentation, and what a fantastic start to this event. As with all the other talks, we're likely going to have lots of questions come in from the audience, and I can see that we have received some already. So I'll move straight on to the Q&A now. So Dr. Plummer, your first question is going to be, what is the biggest hurdle you face in using a new spatial technology?
So in my opinion, my biggest hurdle is being the computation. So really thinking about how are you going to handle these data sets after some of them you don't need a machine for, but after you have all this data, how are you vetting how to process the data, how to store the data, and then how to visualize and interpret the data. So I think there will be, and I have a whole slide for that. But I think that we're making a lot of headway, and I would say that my people have to check GitHub every week, and you'll probably see like 10 new computational tools just for spatial omics. And I think that most people in the field are really thinking about this, so that's exciting.
That's fantastic. Thank you, Jasmine. And just to follow on to that, what criteria do you use to adopt/vet a new sort of technology?
Oh, I think everybody saw a lot of my data across all the technologies. I will say it really has to do with the general need. So these are not, for the most part, they're large pieces of equipment. So I would say there are easy ways to get into spatial where you don't have to buy a capital equipment or use your shared resource for. I would say my easiest way to get into it is usually sequencing based. So most people kind of know they don't need that big push for computation because they know how to handle sequencing data. You could probably get people to help you make the libraries. So I really think some of the techniques like genomics and Visium are a good start to get in the field. How I vet the larger technologies in terms of they're not larger, they're just more capacity building technologies of being able to have the big machine in places to kind of do the computation. Those are really at a level by which biology should be the forefront. And so what are your biological questions? What can you get out of it? If you're an individual PI lab and you love tumors and you're only going to do FFPE, and then there might be technologies that you're really interested in that. But I would say absolutely try with sequencing based first, and then also think about ways by which you can vet publicly available data sets, because it's not all probes. It's like what are the right probes? What are the right antibodies to kind of throw at your assay or your question?
That's an excellent answer. Thank you so much. We have time for a few more questions today, and I suppose to sort of continue on that, what computational pipelines aid you best in your studies? I know you touched upon that briefly during your presentation, but
Sure. So in spatial, there's a lot of tools already out there. So even Seurat, so for single cell genomics, Seurat kind of made a package, I think, specifically for spatial. Geomics has been out for a while, so there are lots of ways by which you can just plug and play. I think Illumina has some ways by which you can use Dragon or other tools online, same with NanoString. So those are easy. That's kind of why I introduced this sequencing to begin with. I will say that some of the other machines already have tools available on them for some of the RNA-ish probes. Most of those are kind of available on the machine itself, but there's usually, that's for the processing. Usually there's some other downstream. I will say that a lot of the tools for the single cell genomics can be co-opted once you get a better idea of what a cell is. So that's the thing, and I alluded to that we're really struggling with, is you can pretty much use any single cell tool as long as you can assign a gene into it, a protein into it, a transcript into it, right? And I will say that requires a lot more mining of the data. I know a lot of people are using things like StarDis and DeepCell. And so I would say that there are things out there make sure your cell segmentation is very good in the beginning and the rest will kind of come.
thank you so much, Jasmine. And we have time for one final question today, which is a great way to round off the presentation, really. Do you really see spatial biology being widely adopted, and why?
Well, I think it already is widely adopted. So I, for the first time as a molecularly trained person, I'm coming from the other end. And so most, having been sequencing a long time, I think most people kind of went from bulk into single cell, DNA extraction by PCR, those kind of things. It's the first time by which pathologists are coming from the back end, right? So not only do you have genomicists from the front end kind of bringing their single cell into spatial, pathology has been around for a long time. It's not just for cancer. I mean, people who investigate Crohn's disease, or you saw pictures of brain tissue, it's that people have always, I mean, IHC, basic immunohistochemistry, exists in almost every lab because people, if you see it, you can believe it. And so I think that's really, we're at the forefront of this explosion of the technology finally being here to ask more cell specific questions, right? Not being reliant on one marker, but those markers have been around for a long time. So now we can just make new biological discoveries into kind of things that we've been thinking about for a long time. I think that's the really exciting part is I wrote a review about how Roman E. Cajal was drawing neurons in the 1800s, right? We've always, as biologists and scientists, and even as clinicians, want to see things and put them in context to where, you know, the disease may start, or what does normal development look like, and why did that drug work? And I think when you can see it in the tissue itself, there's a more believability to how you're going to interpret that and then kind of expand on answering that question.
Thank you so much, Jasmine. Unfortunately, that is all that we have time for, but it has been an absolute pleasure having you present with us today.
Thank
Any
you for
questions?
having me.
No, thank you very much. Any questions that we didn't have time to get to will be sent to the speaker after the event. As with all the other talks in the event, this talk will be available to view whenever you want for the next 30 days. And that just leaves me to say thank you once again, Dr. Jasmine Plummer, and we'll be back soon with the next talk.